art lines. You art line.
Yeah, like so what is section? Section.
This kind of people.
Architecture.
सही है सही है
Three movies I'm really guest. Strike check. I really
Book number 7 and 9 with your mental. Collect your ID card. Please.
it's for sure. Movie so long. So bad after
Like started we will share the video class. Let's see if I feel you having possibility or I so nice. Yeah, one the live stream has started on our YouTube channel. So if you would like to share it with your relatives, friends, so share the links of our YouTube channel. Hello. We would be sharing the link of the live stream in 2 minutes. The live is started in our channel. So if you want, you can. Also, group 7 and 9 collect your ID cards. Yes, I'm sending the links. The YouTube channel, YouTube the country, so you can spawn and link the shot. I think it's important because they're going to be like 5 of being like one year of Just to just one thing. Why? Six, seven. Group 10, talk with your mentors to collect your ID card.
जजिंग मैट्रिक्स उधर ही थी अरे इधर ही थी That's what you normally. Sounds for jazz.
Morning, everyone. Can we have silence and can everyone be seated now? Sorry, time's up for preparing. It's time to start. Requesting all parents to be seated as well.
एक जो पहला स्टैंडिंग
Guys, the judges are now going to enter. So requesting everyone to be seated, putting your cell phones on silent. And when the judges enter, please be courteous to clap and greet them. Thank you very much.
देखते रहना फैसला बन जाना और इसे ऐसे भी घुमाना थोड़ा सुना के बोली वो ज्यादा हाथ और जो भाषा से बोलते तो वो नहीं के भाई
When I asked you to name, I told you give me a report.
यार I mean, चालू बनाने वाले हैं yeah, die from the connected to people.
Google hockey color but my face just what's up now?
Good morning once again. Can you be seated?
Yeah, the seats are coming.
So, good morning, young biologists, future innovators, and welcome to the grand finale of BioQuest 25. Today is not just a conclusion, it's a celebration of young minds who are there to dream, question the ordinary, and journey through science with passion and innovation.
That's right, Arya. After two electrifying rounds, we are officially stepping into the finale of the BioQuest 25. And this round is extra special
Because what awaits is not just a journey, but a goodie of lifetimes, and the finale where we all have come to IIT Bombay.
so before we dive in, let's take a moment to talk about who's behind all this curiosity, innovative, and the science. So of course, first of all, you students are, and the seconds are we are a team iGEM, IIT Bombay, and team iGEM, MIT WPU, part of the global iGEM community.
So, y'all are wondering what is iGEM? To give y'all a brief, iGEM is the International Genetically Engineered Machine. It's a competition. It's like the Olympics of synthetic biology. Teams from across the world use science to solve real world problems and from fun diagnostics to climatic actions and more.
So what we started with was over 1500 students has now led us to this very moment, the final stage, the 60 of you. So you all deserve a huge round of applause. So final stage where our top participants will present their ideas after more than a month of rigorous scientific exploration. So just like iGen, BioQuest encourage students to think beyond their textbooks. work in teams, develop creative ideas, and propose solutions rooted in synthetic biology and towards the biotech.
Throughout the month, students have participated in concept sessions, have given multiple tests, and have deserved their spot here. So, not to mention with honesty, dedication, and creative thinking.
So each team was here mentored by experienced students, us, and field technologists who guided them with insights, encouragement, and feedbacks. So the final presentation that you will see today are not just the answers; they are the outcomes of critical thinking, teamwork, and the scientific curiosity of you students.
So, before we begin, we are deeply honored to have with us a panel of distinguished judges who bring rich experience in science, technology, and education from the Department of Biosciences and Bioengineering, IIT Bombay.
We would now like to invite our panel of judges to present panel of judges to come to the podium and receive a small memento from us. So, firstly, Prof. Swapnil Shinde, you can come on the stage, and Sanvi, you can present the moment. Would you like to take one? Okay. So now I request Professor Rajesh Patil. Now I guess Professor Sanjeeva Srivastava to come on the stage. Thank you so much to all our judges for taking their precious time out to be here. Your guidance and feedback will be invaluable to our young audience.
So, thank you, Professor. So, here's how today is going to go. Each team will have 10 minutes to present their pitches, including any video or animation. This will be followed by 2 minutes or 3 minutes of question and answers from the judges. After the 10th team, we will break for lunch at 1 o'clock and the lunch will be till 1:45. Post lunch, presentations will continue, followed by results and closing by 6 o'clock. Parents are welcome to stay throughout the session. Seating has been arranged. Also, a reminder to please keep your cell phones on silent. The entire event is also being live streamed on YouTube. Feel free to share the link with your friends and family.
So now let's begin the final presentations of the BioQuest and see what happens after the science meets your imagination. So the order in which the teams would present here would be random. So it would go in a way I would announce a team. It would be picked randomly, so you would know. Then you would come here. So first two teams are Team 7 and Team 3. So we would start in 10 minutes. So like Team 7 is ready. Yeah, Team 7, come on the podium with your mentor.
I guess you will work by the time like the first team that extra. But it's not same.
Okay, so our group 7 is ready. So can we have a huge round of applause for the
So, good morning, everyone. We are Team 7 and I'm Shreya. So, today we are presenting our project on a critical environment issue, soil salinity. So, let's start with the introduction. Soil salinity is the major challenge in our country, which is literally occupying the 7.6 million hectare square of area like Rajasthan and Gujarat. So, soil salinity majorly causes the osmotic stress uh, iron toxicity, etc. So, soil salinity, which which is very important to so soil salinity is a major issue which is still like existing in our country and uh, we are still tackling it. So our idea is to promote a sustainable biological solution so that we can tackle it properly with a biological way. So soil salinity majorly uh, decrease our basic crop nutrients like rice, wheats, etc. That are the backbone of our basic Indian diet. So the farmers' income was decreasing. Therefore, we had to tackle it important. So next, we have our existing solutions. So we have two cases. First one is of Team China. Team China has engineered a bacterium which produce gamma polyglutamic acid, a natural biopolymer which reduces the soil salinity, uh, uh, help to uh, help to regain its structure and also lower the pH level. As you can see in the diagram, how the how the poly how the glutamic acid reacting with the soil and making it function. So next we have our IICR Pune, which engineered a Azosphagium bacterium which overexpressed the ACC diamase enzyme which lowered the stress in plants. It has dual benefits like it can make the plant grow uh, under saline condition and drought conditions too. So hand over to Siddharth for the next topic.
Thank you, Shreya. I am Siddharth and I will be presenting our solution. So we have used a bacteria called Chromohelobacter saxeligus. It is a halophilic bacteria, which means it lives in very harsh saline conditions. So also a root colonizer, and it produces proline genes, which help the bacteria stay in these salty conditions. So now you may wonder, how does proline help the plant or the bacteria to protect itself from the osmotic stress? So it majorly does three things. Firstly, it plays an osmoprotectant role, which helps maintain the water potential during the stress. Secondly, it stabilizes the proteins and the membranes during the stress and also protects it from becoming dehydrated. Thirdly, it gives nitrogen and energy after the stress so that the cell or the plant can recover easily. You can see it clearly through this diagram. So we take the genes from C. saccharomyces and put them into a host organism, which is Bacillus subtilis. So you may wonder, why Bacillus subtilis? It is a strong root colonizer. It is also easy to genetically engineer. It is found almost in every soil, in your backyard, in that potted plant, everywhere. It also found very resilient spores, which can survive through almost anything: heat, drought, storage, everything. It is also ideal for carrying salt tolerant genes. So you may now wonder, how do you put genes from Chromohelobacter saccharomyces into Bacillus subtilis? So you basically take a gene strand from Chromohelobacter saccharomyces, we take a plasmid ring from Bacillus subtilis, and then we join them together to make a new plasmid ring, which we now insert back into the bacteria, which is now amplified and made a gene colony. and now we can use it. So to explain this in more detail, I give it to Adyan.
Good morning, everyone. My name is Adyan Gupta, and I will be explaining more in detail about the genetic circuit design. So basically, what we're doing is we have selected the Bacillus subtilis as our host bacteria because it is a strong bacteria which can naturally survive in saline soils. It has this spore formation ability, and it can colonize the roots of the plants very well. So we have chosen the genes Pro-A, Pro-T, and Pro-C because this gene cluster can convert glutamate into proline step by through a step-by-step process. Also, we have used a suitable promoter, which is specifically for you, so that the expression of these genes is controlled and it is regulated. Also, this continuous production will enhance the soil quality and it will improve the growth of the plants and the bacteria. So now about the proline pathway, how proline is made. So first of all, Pro-B, it will take the glutamate and it will convert it into gamma glutamyl phosphate, which is taken up by the Pro-A gene and it converts it into glutamate 5-semialdehyde. And this is converted by Pro-C to P5CR and then to P5C and finally it gets converted into proline. As you can see, this is a 3D diagram of proline. So, why did we select these genes and what is the purpose? So, as discussed earlier, we selected the ProABC genes because it produces proline, and we have used the ProU promoter, which is activated by high salinity. Now, we have used this promoter because it will reduce the metabolic burden of the bacteria and it will also increase the energy efficiency. Uh, as you can see, there is a model plasmid in which we have inserted the three genes. We have the kill switch, the promoter, ORI, etc. things. We have also used the HPH gene as a selection marker so that it provides resistance to hygromycin, which is important as in the lab we can use it for screening. So finally, this model plasmid it will be inserted into our Bacillus subtilis, and this will ensure that uh, the plant or the bacteria both thrive and grow properly in saline soils. This will be a great help to the farmers, especially in salt-affected areas like Gujarat and Rajasthan. So now I'll hand it over to Arundhati for presenting the USP and innovation.
Good morning. I would like to highlight our unique selling point or our innovation in this. It comes out in three major aspects: our use of proline, our quorum sensing in the form of kill switch, and our deployment strategies. So, as my teammates mentioned before, proline is an excellent osmolyte, which means it balances osmotic pressure in plants. So, even in saline soil, they're able to efficiently absorb water. Now, it's also an antioxidant and it helps to plants to absorb phosphorus and nitrogen. So, in our quorum sensing, it's a well-established mechanism in the subtilis where each cell secretes COMX, a peptide, which allows them to understand the size and extent of their population. Now, we found out that if we use this mechanism to activate COM-A, a regulatory enzyme, it could in turn activate a promoter, PCOM-G, to induce the expression of MAS-F, a toxic gene. This will ensure that there's no overpopulation of B subtypes and it will kill the cells and bring it to the core or the exact amount of B subtypes required. Now, this is extremely important to make sure that proline is not excessively secreted in soil. Now, for our deployment strategies, the first is the quite industrially mass-producible one of sodium alginate beans. This is basically seaweed extract solidified by calcium chloride. Hence, it's very eco-friendly and it can be easily produced in industries as mentioned before. And for our very innovative fungal seeder, so these consist of a fungi like pea also, which will make sure that our engineered pea subtilis stays in soil for long periods of time. It consists of vermicompost extract to make sure its integration in soil is very easy. These two ways will ensure our success in commercialization, which is what Samyaka shall explain next.
Good morning. I'll be explaining our commercialization strategy. First, proof of concept. Why do we need a proof of concept? We need a proof of concept to check if our engineered bee subtilis works effectively when encapsulated in alginate trichlorose beads. We grow the bacteria, form beads, and test them in both saline and normal soil. We measure the protein levels and plant growth to check if our engineered to check if our concept works effectively. Second, prototype and pilot production. After lab success, we make 1 to 5 kg of uh, we make 1 to 5 kg of smart biobeads. We then grow bulk cultures, form beads, and package them for trial testing. We then use them in small trial plots or by local farmers. Third, startup and legal setup. What are the first steps to legally launch our company? We register under MCA and apply for India Startup. We then trademark our company and company name and product and get IBSC and RCGM biosafety approval. Our basic setups also include GST, Udyam, etc. Fourth, manufacturing and distribution strategy. Our manufacturing of beads will occur in pilot facilities with food safe alginate. Packaging will be done in rupees 5 biodegradable sachets for easy use. Distribution will occur through Krishi Kendras, vans, etc. Fourth, fifth, fundraising and grant strategy. We'll apply for BIRAC Big, Nidhi Prayas, and DST seed grants. CSR support from Tata Trust and Reliance may also help. Accelerate generators programs will guide us through strategy and funding. Sixth, regulatory and safety plan. Since we're using GMOs, biosafety is a very essential aspect. We follow the IBSC and RCGM biosafety rules and we use skill switches for control. Environment testing helps us like to know whether our concept works in real life or not, whether it is safe or not. So thank you. I'll be handing over to Jenny.
Biosafety. Biosafety. In conclusion, a solution offers a safe and smart way to ensure soil salinity, protect plants from soil salinity. We use a special gene from seeds, salinity genes, and added to Bacillus subtilis. It helps to overcome soil stress. It helps in growth of roots, plant roots, helps in using minerals and water wisely, and is always used where there is salt present in soil. The fertilizer bombs is used can be used to apply in large fields, and the kill switch used in the bacteria ensures safety. Um, to share your idea creatively, we have made a booklet as a creative aspect.
Thank you, guys. It was a lovely presentation. Judges, do we have any questions for the group?
All right, so very nice presentation. And
we took very systematic approach of thinking how to look at or expressing the protein, and then thinking about how to elevate the salt salinity of. But just think about in the real field when you have to impart that on the plants, right? Don't you think like the let's say bacillus being bacteria, if you want to try it out in the next level, should you try out in some model organism in the plants to look at the protein expression and how your overexpression is working or not working?
Yeah, so there are many salt indicator plants such as tomato and rice, which will show like when the soil salinity is high, and then we can do a lab and we did a model experiment also, and it will be safe for the real.
Right. How you will ensure that in the bacillus when you're overexpressing, right? What is the level of proline expression that you have? How will you do that? So you have you talked about the quorum sales. So how will you actually measure the expression of
So we're focusing on the
what will be the rough approximation?
it's actually already established, so there must be a limit where each cell understands that it's over the population, it's over the whole. So when it kills the cells, it ensures that more cells are not able to produce. But it doesn't ensure that.
Experimentally, how you will monitor that your cloning and overexpression strategy has worked well, and what is the cloning expression happening in the bacteria?
It's already there are many research papers which show how colons are.
How? No, not what I'm saying. I'm just saying, think about it. When proline you are overexpressing, maybe it is possible that your proline strategy has not worked out well, and proline expression is not sufficient, or maybe it is optimal, maybe it is very subtle. How you will measure that the expression is optimum or not?
Well, like I said, there are tomato and rice plants will change when there is optimal salinity in the soil. So if there is more or less, like the leaf on tomato plants, the leaf will turn yellow if there is too much soil, and if it is too less, there will be stunted growth. So by that we can see whether our soil is more, less, or right.
It's very indirect comparison. So you have to first look at more biochemically how you can establish the expression level, and then the plants will be indirect indicators. It might affect from many of the environmental factors as well. It cannot just directly attribute to the only coding level, right? You to also establish experimentally how you can measure how much you are expressing. And of course, eventually, like you might want to look into some other model plant systems as well and see if that expression working well or something. But overall, we'll see. This question is from all of our judges, so to save time, we are not just going to ask one by one. We are just going to have one question for all. Right.
Thank you very much, guys. Can you have group ready? So what the group salinity, crop salinity that your group spoke about was very insightful, and we are all very excited to have next group.
After this presentation, it would be group 10's turn. So be ready with your pen.
Yeah.
Hello, mic check, mic check. Hello. Are we ready, guys? So all the best, and this is group 3. What is the bacteria?
To kill cancer, to kill and kill tumors. What if we could train our bacteria to hunt cancer, kill tumor cells, and to destroy itself before reaching any part of the body? Sounds like sci-fi, right? We made it here. Meet Salvo X. Good morning, everyone present here. You all might know about cancer, but do you all know what happens in cancer treatment? In cancer treatment, one of the biggest challenges is to get drugs to attack only the tumor without harming the healthy parts of the body. Many current treatments spread everywhere, leading to harsh side effects and limiting how much medicines patients can safely get. To tackle this problem, we have found out SalvoX, a genetically engineered Salmonella typhimurium bacteria. This approach offers hope to fight cancer more precisely. So, so the problem statement for our project is delivering therapeutic agents specifically to solid tumors while minimizing damage to healthy tissues remains a major challenge in oncology, often leading to systemic toxicity. Thank you.
Cancer is one of the major health problems faced by humanity ever. Despite years of progress, still millions are affected each year. Common therapies are chemotherapy, radiotherapy, and immunotherapy. But these therapies include radiation, which kill the cancer cells, but also damage the healthy tissues and cells. The side effects of these include nausea, headache, and long term tissue damage. So, a solution is required which is both kills the cancer cells and is precise, which does not harm the healthy tissues and cells.
Now, there are some existing solutions. Now, let's go through them. First is Sign Logic SYNP1891. What it uses is a friendly E. coli strain that wakes up your immune system in the body. But it was missing. But there was one thing that it was missing, that it only uses one trigger and does not release a toxin, kill cancer directly. It's like kind of going to a war with but without any weapon. Second, Ernest Pharmaceuticals. What it uses is a modified salmonella that self-destructs after giving helpful proteins to the tumor sites. But what it was missing, that it doesn't use a smart nanoparticle like we do, but the bacteria, so that the bacteria might not break open too soon. So not as targeted study ready. But the third one, Vaccine GmbH, uses a Salmonella T521A carrying VEGFR2, the target in blood vessel, but and activates T lymphocyte cells to attack these vessels. It is being tested in brain cancer, glioblastoma, but it was missing. There was something missing. That was it mainly helps immune cells and not killing cancer cells directly. This makes Salvo X one of the safest and smartest ways to treat cancer with bacteria today.
We have looked at the problem, so now let's look at our solution. Our solution is SalvoX, which is a genetically engineered Salmonella typhimurium.
Next slide.
So in Salmonella typhimurium, we will include the plasmid PACYC184, which has low copy number. Low copy number ensures that there are less copies of the plasmid present inside the cell, so that cell does not get overwhelmed by the number of by the number of by the large number of plasmids, and it also ensures that cytolysine A is not produced in excess, as it may then harm our normal tissues as well. So cytolysine A is our therapeutic gene which will kill the tumor cells. So it will only get activated under the tumor microenvironment. This is because we have two promoters, NARG and ASR. NARG is a hypoxia responsive promoter, and ASR is a acidic pH responsive promoter. So both of these conditions are provided by the tumor's microenvironment. Hence, cytolysine A will cause tumor cell lysis. After that, once the tumor cell lysis occur, sorry, after that After that, as cytolysine A occurs, cytolysine A causes tumor cell lysis. We use P-SOX. We have promoter named P-SOX-S, which is a kill switch promoter. So once tumor cell lysis occur, our hypoxia environment will get reduced, and it will again have oxygen levels stable, and acidic pH will become neutral. Hence, a PSOX-S, which is responsive to oxygen, will get activated, and hence lysis protein E will be read by our promoter PSOX-S, which will allow the bacteria to self-terminate itself, ensuring that the bacteria and the ensuring that the bacteria and the tumor are both eliminated. Next slide. So we have looked at how we will kill the tumor cells. Let's look how we will transport the tumor cells, how we will transport the salmonella typhimurium to the tumor cells. For that, we are going to use dual protective layer, which is above layer is the polyketal and the inner layer is ziphate. Both will provide protection to the salmonella typhimurium in the circulatory system. And when it is under the tumor, when it reaches the tumor, both the layers will get degraded, and hence our bacteria will be released, and it will do the killing. Next slide. So this is just summarizes what I just said now. Next slide. Over to Amol.
The unique selling point of our project is number one, it only works in tumor specific microenvironments like low oxygen levels and high acidity levels. Then, second, it self destructs in healthy tissue. It has a kill switch lysis protein E to stop and destroy itself outside tumors using yes gate. Number three, it has a safe delivery. Uses biodegradable pH sensitive polymer that degrades in acidic microenvironments. Next slide.
Now, this is the research methodology. First, we identified our problem. Cancer treatment in India and around the world harms healthy cells. So, we needed targeted and a smart therapeutic system. We choose Salmonella typhimurium, which has a tumor homing ability and thrives in hypoxic and acidic microenvironments. Then, the therapeutic gene selection. We chose cytolysine A, which can be obtained from Shigella or Salmonella typhi or paratyphi. Cytolysine A forms tiny pores or holes in the tumor cells and causing them to burst or cause cell lysis. It has a strong toxic effect and that helps kill tumor cells effectively. The logic gate design, tumor specific expression and gate logic, as Vihan explained, we use Naji promoter that activates under low oxygen and ASR promoter that is activate under acidic pH. Hence, when both are activated, cytolysine A is expressed. Safety mechanism, the P-SOX promoter turns on in oxygen-rich areas like healthy tissues, and it activates lysis gene E, causing the bacteria to self-destruct outside the tumor, which keeps the system safe and controlled. Then is the plasmid construction. Backbone of our bacteria is the PACYC184, the plasmid, and tools such as restriction digestion plus T4 DNA lipase. Assembled parts like others, promoters, toxin, gene, kill switch in silico validation. This is very important. We use lab exchange and IIT-B simulation to simulate our experiment at specific sites. Carried out DNA ligation to join designed DNA fingerprint fragments and inserted the microbinant plasmid into salmonella for further expression. Targeted delivery system. We used pH sensitive polyketal nanoparticles to carry the engineered salmonella and which is stable in bloodstream, protecting bacteria during circulation. Breakdown in acidic tumor environments, releasing salmonella at the target site. Ethical and safety evaluation. Environment sensitive promoters for controlled experiments. No antibiotic resistance is released from built in self destruct, biodegradable content delivery system, and no release to open environment.
Last 2 minutes.
Proof of experimentation. So we basically simulated our full expression workflow of Salvo X using E. coli DS5 alpha for cloning and BL21 for protein production. Our goal was to confirm the successful expression of our key genes, cytolysine A and lysis E. And our processes followed these core steps. Next slide. So, bacterial growth and plasmid construction. We grew DS5 alpha at 37 degrees Celsius for 24 hours and then harvested the cells via centrifugation. And our plasmids carried cytolysin A and lysis E under promoters which are responsive to tumor triggers. Next slide. So this is basically our transformation and selection. We made the cells chemically competent, and then we used heat shock at 42 degrees Celsius for plasmid uptake. And all the transformed cells were plated on antibiotic agar, and only the successful transformants formed colonies. Next slide. Induction and lysis. What we basically did here was that we introduced our plasmids into the BL21 proteins for protein expression, and then we induced with IPTG, mimicking tumor conditions. And then finally, we harvested and lysed the cells to collect the proteins.
Salvox, smart bacteria for smart cancer treatment. Cancer treatment in India is either too toxic, costly, or inaccessible. Salvox uses engineered bacteria to detect and destroy the tumor from inside of the body. Marketing, targeting 1000 to 1005 crores in India's 16,000 crore cancer market, starting with breast, pancreatic, and colorectal cancers. Business, just 5 to 7 lakhs per therapy, which is 3 times cheaper than CART, plus gene circuit licensing. Risks, we ensure biosafety with kill switches, fast track approvals via India's biotech policy, and address GMO through transparency and data. Roadmap, preclinical trials complete by 2026, human trials phase 1 and 2 in 2026 to 2027, and commercial launch by late 2028 in partnership with Indian hospitals. Impact, early detection, precision therapy, biotech jobs, global IP, and affordable access via government schemes like Ayushman Bharat. Backed by ICMR, CDSCO, and DBT, it is India made and world ready. Thank you.
Time's up.
So we have made 2 websites for this project. So this basically is an interactive website that briefs everyone on the basically all the necessary information about our project. This is the second website. SalvoX isn't just science, it's intention. Every single part of our system has been chosen to solve a specific problem. From our kill switch, which ensures self destruction outside the tumor sites, our logic gate that only fires in acidic and hypoxic conditions to ensure safety, and our smart layered nanoparticle, which is designed to release, evade, and protect. We are Salvo X, and we take pride in precision.
All right, so good project, very thoughtfully done. Uh, I think this approach essentially is going to distinguish between healthy and the tumor, right? And your goal will be to look at more when the hypoxic condition is there and acidity is there. Then going to target more about the cancer cell. And in the healthy is going to do very minimal impact per se, right? Or going to get self-destructed. Now think about the early stage of cancer when the cancer growth is not too much, right? When the cancer advances in the grade 2, 3, or in the case of brain tumor, glioblastoma, very advanced. I think this approach might be more useful at that point because now you have a lot of tumor cell and you have so much proliferation, and now too much hypoxic condition. It's very easy to target. But in the early stage, when you have very little tumor cell, how are you thinking that your technology will be helpful? Did you understand the question?
Yes, I did. I did. So, yes, while we haven't taken the early stage into consideration, we also have to consider the fact that in India, most of the patients, so like when cancer is detected, it is usually detected in the later stages, which is why our solution targets all the symptoms that would show up in the later stages. And actually, I think around 90% of the cases are very late detections, while these conditions have already been present. So, which is why we use the dual triggers to target acidic and hypoxic environments.
Any specific reason why you chose pancreatic, colorectal, and breast cancer? And
Yes, because these are anyone else wants to answer? Yeah, so these are very surface level tumors, and our design right now, it is basically, although it can also reach the deeper parts, it's not that it can't, but delivery is much easier for all the surface level tumors, and these are very surface level, right? So which is why it is more suitable for these right now, but we can definitely also modify the design to make it suitable for different types of cancers.
we did not talk about much about a nanoparticle strategy, especially for this specific thing. Do you want to comment on that?
So that is, huh.
So we are using, so the nanoparticle has two layers. First, the polykettle and ziffate. So polykettle is a biodegradable polymer. So what it is also very like neutral to our body. It does not harm our body. So we are using it for so that the salmonella bacteria can go to the tumor cell without being damaged in the blood circulation, like by the sense of immune system or anything that may cause it harm. Zifid. So Zifid we added because Zifid blocks the oxygen. It blocks the oxygen to enter inside it. So we have a kill switch, right? Which activates under oxygen environment. So we do not want the oxygen of blood of the circulatory system to give an early death to our cells. That's why we added Zifid. Thank you.
Can we have group 10 ready? So a huge round of applause for group 3 tackling something like cancer and the thought process they put into everything. Next group is group 1. Please be ready with your mentors. Thank you. Group 1, please be ready.
Time management are going there, but bioethics is why none of them are able to talk much. Yes. Wait. So it's a problem generation. Yes. Bioethics part, they're not able to talk much. So I don't know how to bring it for now. Sir, it's not enough to explain. But nevertheless, if you are keeping one point of bioethics, they should speak out some way of, otherwise that becomes very general period. But I'm saying it could be very unfair, there's got to change the dynamic. We can adjust the so or maybe even the three policy, the bioethics we can drop. And novelty we can make it why better. Variety Yeah, now it is not there in the biopics to the finding and you select one of them from that slide, it becomes if you give the mark of the change. Second, another thing which we biopsy on then speak of
Some of the teams that can say stuff use it. Some of the teams will be like,
something of this should or not have and it will not be given
or intelligence rank to the scale of very good.
just not trying to get out the
One of the teams.
Then laptops.
then go to work.
So you can give
Change was
Flight streaming.
all the guys.
Yeah.
Good morning, professors. Today we are going to be talking about snake venom. Uh, every year, around 5 million people are affected by snake venom. This isn't just a number, it's a crisis. Globally, 81,000 to 138,000 people die every year because of snake venom. In India alone, this figure is about 50,000 deaths per year and even higher in some estimates. Uh, only the existing solutions, which are the traditional antivenoms, are failing us. Only 74.4% of the people who are affected receive these treatments, leaving about a quarter of people vulnerable to it. Even worse, 7.5% of the people who are treated are report adverse side effects due to antivenoms. And an average person receives only 4.5 vials of antivenom when 7 vials or more are likely required. There are only 46 producers of antivenom throughout the world. In Africa, for example, Africa receives only 2.5% of the antivenom which it actually requires. This is not just about snake bites, it's about a preventable disaster. Our solution, Venom Shield, is poised to disrupt these markets with a new generation, affordable and safe approach. So, who's ready to save some lives?
We are presenting to you the Venom Shield.
A Venom Shield is an affordable and accessible neurotoxic venom based. It targets neurotoxic venom with cutting edge biotechnology. It uses genetically modified nicotine acetylcholine receptors. Receptors act like magnets for snake venom, quickly bind and neutralize the venom. They do not affect the normal acetylcholine signals, which enables fast and targeted protection.
So before we start, we need to understand the basic and normal functioning of the muscle. Muscles have certain receptors known as nicotinic acetylcholine receptors. These receptors, when they bind to acetylcholine, they induce a conformational change which opens up an ion channel and therefore the muscle contracts. Now, the problem is that when the neurotoxin enters the bloodstream, this neurotoxin itself starts binding with those receptors, therefore, and it has a special property that it does not induce the conformational change. Therefore, ion channel does not open, the muscle does not contract, and a person can suffer paralysis. And the worst part of it is cardiorespiratory arrest can also happen. So we'll be understanding some factors regarding binding to receptors. Hydrophobicity is very important. Many neurotoxins in general contain hydrophobic ends.
These hydrophobic ends would prefer to bind with hydrophobic binding sites of the receptor. We can say that higher hydrophobicity of the receptor would increase affinity to the neurotoxins. Hydrogen bonds are bonds between electronegative atoms and hydrogen. These bonds provide strength to the particular bond and make the protein more stable. Enhancing hydrogen bonds increases toxin affinity. Cation pi interactions are interactions between cation, which is acetylcholine, and electron cloud of aromatic ring within the receptor, which is triosine. If we disrupt these interactions, the acetylcholine bond to the receptor would weaken and comparatively the toxin affinity would increase. Electrostatic complementarity means that if there are two ligands, A and B, one is positive, the other one is negative. This enhances the strength of the bond bind, and a specific property is that the neurotoxin would bind to the receptor and not to any other molecule receptor resembling the binding site. So our goal is to create decoys that basically bind the venom toxin to itself. So to increase the venom affinity of these decoys, we have, after an extensive literature review, we have identified some of the mutations and they are as given. After talking to an expert, Mr. Arhan, we have confirmed mutations at 113, 171, 210, and 218 in triosine and change it to non-aromatic amino acids such as lysine.
Five more minutes.
This is the structure of NaCHR with alpha, beta, delta, and epsilon subunit. This is the alpha subunit. The human body has 10 types of alpha subunit. The, the yellow part which you can see in this is the acetylcholine. This is the model in which the acetylcholine is bound.
So we cannot directly change the proteins. We change the DNA that instructs the particular cell to make those proteins, and therefore we introduce our proteins in that way. So first we have to identify our target residues, that which amino acids are responsible for the binding. We use high resolution NACHR images and we use computational docking to map these structures. In the second step, we design mutagenic primers. The word primer is self-explanatory. Prime means to start the process. It starts the process by separating the two strands and then it binds to it. Some conditions such as temperature greater than 78 degrees Celsius is required for separating strands and GC content is required for introducing hydrogen bonds to make the structure more stable. Site-directed mutagenesis, we use PCR polymerase chain reaction with mutagenic primers. We are basically now duplicating the gene that we have made and we put it into a plasmid for modification. In the fourth step, we are extracting the mutated DNA from bacterial cultures. We confirm mutation with Sanger sequencing, which is also known as chain termination sequencing. Then we inject the plasmid into HEK293, which are embryonic kidney cells. These cells show more expression and are easier to study. After that, we have to verify the mutation. We are using Western blotting, which is separating the mutations by size, placing them onto a membrane, and then checking that whether the desired mutations have been done. Immunofluorescence is nothing but using antigen and antibody interactions to find out that whether that particular protein is present in the receptor or not. After that, we have to perform functional testing. We are using radioligand binding, which are substitutes for actual toxin. We are using radioactive materials that resemble toxins. Another method is to use a two electrode voltage clamp And the principle behind it would be that if acetylcholine binds to the receptor, the voltage will be generated.
Used for 120 years, the existing antivenom has many limitations. Some of these limitations are listed here. So, what's our unique selling point? First, we are developing a game-changing next-generation antivenom vaccines. These are our solution is recombinant human antivenom, which are lab-made human antibodies which are completely safe, work quickly, and they show no allergic reactions because they are not made from animal products. Plus, it's heat stable, meaning there is no need for cold storage. That is perfect for areas perfect for remote areas where it's needed the most. Imagine rapid neutralization happening in your bloodstreams with the antibodies and stopping the venom before it spreads. This is preventive protection training the immune system before the bite, unlike the traditional antivenom, which is given after the bite. And we are designing it to be affordable and accessible. These are produced using low cost, fast biotic methods, which specifically in the regions, high risk regions like Africa, India, and Southeast Asia. Also, another unique selling point for our vaccine is that it does not use animal products, unlike the traditional antivenom, which is produced using animal antibodies.
We are collaborating with Serum Institute of India, Bharat Biotech, Biological E Limited, etc. These are the some companies which are experienced in biotechnology and will help us in our success. Another, the cost and pricing. The cold storage costs are minimal, ranging from rupees 15 to rupees 5 to rupees 15 per dose. Manufacturing each dose is around estimate at rupees 500 to rupees 2000. We are ensuring high quality production while keeping the expenses reasonable. The vaccine will be sold to hospitals at around rupees 1000 to rupees 3000, and we will also sell it to the government by using bulk purchases ranging from rupees 800 to rupees 1500 per dose. This pricing structure will help us in widespread accessibility. There are many possible risks, such as muscle or allergic effects. We are ensuring that these can be solved by clinical lab testing, such as lab and by doing lab and animal testing.
We have conducted a SWOT analysis to know our project. Our strengths include region-specific and highly effective solution against neurotoxic venom, affordable and accessible pricing range. We have used advanced biotechnology and our solution is highly soluble. Our weaknesses include complex and lengthy development process which might take up to 5 to 10 years. High research and development and manufacturing cost, potential risks such as muscle and nerve effects and allergic reaction. Our solution requires a cold chain logistic for storage and distribution in case it needs to be transported in cold storage. Opportunities. Large unmet need in India and globally high due to high snakebite rates. Government support and R&D funding. Potential for inclusion in public health programs and expansion to other venomous threats, regulatory hurdles and approval delays, com- competition from established anti-venom producers, evolution of venom or resistance over time, and public concern about genetic modification technology and safety.
The Indian government has created a dedicated rupees 20,000 crore corpus for the research, development and innovation RDI scheme, which is specifically aimed at encouraging and supporting private sector R&D, including startups and industry-led research. The research and development would take around tens to hundreds of millions of USD over several years. Time's up. Our vision. Let's not just imagine a safer future. Let's build it. With Venom Shield, we can turn the tide against snakebite deaths and offer hope where it's needed most. This isn't just about a product. It's about saving lives and building a healthier world. We invite you to slither with us on this crucial journey. Thank you.
Hello, nice presentation and nice idea. So the question is, are you targeting a particular venom issue or is it applicable to all different types of snake bites?
Yes, it is mainly focused on neurotoxic venom.
So, we are targeting neurotoxic venom by creating decoy receptors so that the neurotoxic venom binds to it. This is applicable for all general types of neurotoxic venom. There are specific types. For example, certain venoms have negatively charged ends, but most of them are positively charged. So we have generalized our methods. No, antibody is not a decoy. We are creating fake receptors. We are creating fake nicotinic acetylcholine receptors so that the neurotoxin present in the venom binds to those fake receptors, because if it binds to the real one, then you can suffer paralysis.
So, initially, like you mentioned, you will do the quality check and you Western blot. What would you expect on Western blot?
You would expect you act in Western blotting, you basically arrange the proteins by size and you transfer them onto a membrane. Then using techniques such as low angle X-ray diffraction or electron microscopy, you identify the particular mutation and then you sequence them. That are they in the correct order? If at what position which mutation is present?
With respect to your receptor, what if it misfires? Like if it's taking something that
Pardon, what do you mean by misfire?
misfires in the sense if it is some irrelevant protein in some other source.
Mm, That won't happen because it has specific mutations that we had made for this purpose only. Acetylcholine.
There it will work for sure.
Yes.
Being or the molecule can also bind to your people.
So it will only hamper the functioning. There will be no negative side effects. Since that receptor is a decoy and it is not attached to your muscles, so anything which is binding to it, it is not relevant to your body functioning. So it will only hamper the function, but it will not produce any negative side effects that could hamper the normal functioning of the body.
Okay. Thank you. Thank you.
Thank you.
Can we have group 10 with the mentor? And next presenting will be group 1. So please be ready with your mentor. Thank you. Right. Now we will have group 1 and then we will have group 6. Thank you. Sorry for the problem.
Can we guys please put 10 minutes for this? 2 minutes flexibility in one go. Like you find somebody. Awesome. I'm being one friend. Yes, yeah. The timing of the remote 10 minutes. Then you can do it from there.
Good morning, everyone. We are Team Core Revival. Heal the reef, end the grief. Coral reefs are the most important aspect of the marine ecosystem. They are also called the rainforest of the sea. They support less. They cover less than 1% of the ocean floor, but support over 25% of the marine life. But now they are bleaching. It is very problematic. They are bleaching due to ocean warming and acidification. All the things are happening because rapid industrialization. India has 5,790 square kilometers of coral reefs which are on the verge of dying, especially in Andaman, Lakshadweep, and Gulf of Mannar. This isn't just an environmental issue, it's deeply human. According to National Oceanic Atmospheric Administration, 84% of the corals are already dead or dying. They live in a symbiotic relationship with an algae called zooxanthellae. When they face heat stress, they expel this algae from their tissue. Just a 1 to 2 degree rise in the temperature, corals start to bleach. Oceans also absorb 30% of the carbon dioxide, which causes acidification, which also decreases their temperature, which also decreases their pH from 8.2 to 8.1. This seems very little, but it is very mass. Now we are seeing how the corals bleach. Look at this beautiful architecture of the sea, how they are bleaching. Now my team will guide you all the way to our project.
This is a very grave problem, but not many people know about this. So we conducted a experiment and tested how many people know about this through a Google Doc. As you can see the stats, not many people know about this. So we took it in our hand to educate the youth by conducting educational outreaches in Pune, Tamil Nadu, and Mumbai.
So now let's take a look at the present solutions. First are the genetically modified zooanthely. They are symbionts created in the lab at thermal heat stresses. So when put in corals, they offer higher heat resistance. Next are assisted gene flow. They are coral crossbreeds. Basically, we take corals from warmer and cooler coral reefs and make them, and the resultant offspring has 26 times higher the heat resistance. CRISPR is a new coming technology that aims to use genetic engineering to emphasize and amplify the gene pathways. Next slide, please. So, now we know about the present solutions. Now, I present to you our own solution, our novel idea, that is Symbio. We have created a new team of three: coral, algae, and bacteria. Bacteria, that is Exigobacterium. They imagine the coral like a phone, the bacteria like a cooling system, and the zooanthely like the battery. When the phone gets heated up, the battery gets heated up too. It drains out and the phone becomes dead. If there is a cooling system like the Exigobacterium, it will cool the battery down and make the phone run smoothly. Next slide, please. So, there will be a question in your minds. Why did we choose this bacteria specifically? Well, Exigobacterium occurs in Yellowstone hot springs at temperatures around 70 degrees Celsius. So, it is a thermophilic bacteria and is naturally heat resistant. The main characteristics that promote all of these characteristics is its gene circuit, which we'll learn about in the next few slides. Now, it is also easily engineerable. It is environmentally friendly and coral adaptable. So let's look at each component of a gene circuit. First is the heat activated promoter, PHSP. It is like, just imagine how in a taps we have sensors that detect our hands and allow the water to flow. Same way, this acts as a sensor for the rising temperatures. When detected, it allows the gene to work, and the protein encoded inside it is shown. DNAK and GROEL. These help the coral proteins that are misfolded to correctly refold and prevent most misfolded proteins too. The SODA, which is super dioxide dimutase A, it treats it actually treats the ROS, basically, which is the reactive oxidative species that that are byproducts that build up in coral during bleaching. SODA prevents oxidative damage. Over to Uddhav.
Okay, so let's imagine a coral cell getting stressed up. Just like a hospital during crisis, the patients for flooding up. Here, the folded proteins are coming in. So we have DNAJ, the nurse. So it spot the damaged protein, bring them to the doctor, which is the DNAK. So here, it brings up the substrate to the DNAK, and then the DNAK has ATP in it. So when ATP allows the substrate to open up and then accept the protein. So after the substrate joins the system, the ATP is converted into ADP, which is adenosine diphosphate, which closes the pathway and locks the patient in the room. So after this, we see group P. So this has a critical. It has a critical.
5 minutes are up.
Okay, it has a critical job in our system. It is just like a hospital chairman. So it facilitates the progress. It removes ADP from DNAK and ensures that DNAK has access to all other the extra folded proteins. So during heat stress, these group B works intensely and makes the equals the number of growing folded proteins and work of DNAK. Next slide. So yeah. So we have our idea of a gene circuit, but how do we prove it or check the quality? So we do that with the help of E. coli. Why E. coli is it is easy and fast to engineer. The process is like we contain DNAK. I mean, our gene circuit contain DNAK, DNAJ, and group E, and we'll build a synthetic gene circuit in E. coli, and then it will be controlled by a heat sensitive inducible promoter. We'll glue the E. coli at a temperature of 37 degrees Celsius to 42 degrees Celsius. We'll activate the stress genes and test the ROS level, which is the reactive oxygen species. So with great biopower comes a great responsibility. To prevent the uncontrolled growth of our bacteria outside the coal environment, we designed a genetical kill switch mechanism. So we have the promoters called keep cons. It's a constitutive promoter that continuously drives the expression of the kill gene. So we have this toxin gene called CCGD. So it encodes the proteins. It stops the replication of the bacterial DNA, sometimes causing the death. So this fusion mechanism ensures that this circuit works only when it's needed, only when the coil is stressed out.
Last 2 minutes.
So yeah, let's see about the regulatory landscapes. Before implementing our gene circuit, we need some functions and guidance and approval from agencies. Those are CDSO and all those things, and USA, we have EPA and MPO. Yeah. So in upcoming slides, we'll discuss about the ethical, social considerations and public stakeholder engagement. Over to Wade.
As we now know about the kill switch, we will continue with the deployment. For deployment, we use biodegradable gel beads that allow the controlled release of engineered microbes into coral reefs. While effective, this poses many challenges such as limited diffusion and earlier breakdown, which is premature bead breakdown, and also potential coral dependency. We also apply the refresh method, where juvenile corals are inoculated with these engineered microbes to improve their growth and survival. This approach, however, raises ethical concerns about favoring engineered corals over the wild ones. To support our coral protection strategy, we conducted a molecular experiment to extract and express heat resistant proteins from Exigobacterium, enabling us to produce them efficiently using E. coli as a host. Gene extraction. Exigobacterium was cultured at 37 degrees Celsius and purified using proteins and RNAs. Gene cloning into plasmid, cut using EcoRI. Transformation into E. coli, in which competent cells were produced using calcium chloride.